Welcome to G Fun Facts Online. Today, we're diving deep into a war being fought on a microscopic battlefield inside the human body. It's your immune system versus cancer. For years, we've been trying to tip the scales in this battle with a revolutionary approach called immunotherapy, which arms our own natural defenses. But cancer is a clever enemy. It uses tricks to evade and even shut down our immune cells. A key battleground is the tumor microenvironment, a hostile landscape where our immune cells struggle. But in this cellular drama, a new hero has emerged, the mitochondrion. You probably know it as the powerhouse of the cell, but it's so much more. It's a central hub that controls metabolism and signaling, and it's the key to making our immune warriors, particularly T cells, more effective. Today, we're exploring the incredible new frontier of bioengineering these cellular powerhouses to revolutionize the fight against cancer. Let's start with the special forces of our immune system, the T cells. Think of them as an elite fighting force, capable of hunting down and destroying cancer cells. Like any elite unit, their performance depends entirely on their energy supply and overall fitness. This is where mitochondria takes center stage. The metabolic state of a T cell, which is directed by its mitochondria, is directly linked to its function. Different types of T cells have different energy needs. First, you have naive T cells. These are soldiers in a standby mode, waiting to be activated. They rely on a very efficient process called oxidative phosphorylation, or OXPHOS, to generate energy from nutrients like glucose and fats. It's a low-power, high-efficiency state, but when a naive T cell encounters a cancer cell, everything changes. It undergoes a dramatic metabolic reprogramming, switching to a process called aerobic glycolysis. This is like hitting the afterburner. It rapidly generates energy and provides the raw materials needed for the T cell to multiply into a massive army of effector T cells. These are the frontline soldiers that directly kill cancer cells. Their mitochondria even change shape, becoming fragmented and punctate to support this high-energy, rapid growth state. After the initial battle, a small group of these T cells become long-lived memory T cells. Their job is to provide long-term immunity and prevent the cancer from coming back. To do this, they switch back to a more sustainable energy model, relying on fatty acid oxidation and the highly efficient OXPHOS again. Their mitochondria also change, fusing together into long, elongated structures that enhance their ability to survive for long periods. So, mitochondria aren't just batteries, they're also command centers. They produce signaling molecules, like reactive oxygen species, that help regulate T cell activation. The very shape of the mitochondria, whether they are fused together or broken apart, helps determine whether a T cell becomes a short-lived killer or a long-term memory cell. The health of these tiny organelles is absolutely critical for a strong and lasting attack against cancer. Now imagine sending these elite T cells into enemy territory. That territory is the tumor microenvironment, or TME, and it's a metabolic war zone. It's a chaotic ecosystem of cancer cells, blood vessels, and other cells, and it's incredibly hostile to our immune warriors. First, it's often hypoxic, meaning there's very little oxygen because the rapidly growing cancer cells use it all up. Second, there's extreme nutrient depletion. Cancer cells have a ravenous appetite for glucose and other nutrients, essentially starving the T cells. Third, the environment is acidic. The cancer cell's high-energy metabolism produces a lot of lactic acid, which lowers the pH and harms T cell function. Finally, cancer cells can release immunosuppressive metabolites, chemicals that actively shut down T cell activity. This brutal environment takes a huge toll on the T cell's mitochondria. They show signs of dysfunction, with decreased mass, impaired energy production, and excessive damage. This metabolic exhaustion is a primary reason why our immune system often fails to destroy tumors. But here's where it gets even more sinister. Recent research has discovered that cancer cells can actually hijack mitochondria directly from T cells. They form tiny physical connections, called tunneling nanotubes, and literally steal the powerhouses from their attackers. This act of mitochondrial theft not only boosts the cancer cell's own energy, but further weakens the immune response. This discovery has made it crystal clear that we need strategies to protect and enhance the mitochondria of our T cells. This brings us to the exciting field of bioengineering. Scientists are now developing innovative strategies to boost the metabolic fitness of T cells, essentially creating super soldiers. One of the most fascinating approaches is mitochondrial transfer. It turns out that some cells in our body, like bone marrow stromal cells, can naturally donate their healthy mitochondria to other cells, including T cells. Inspired by this, researchers are now supercharging T cells by giving them a fresh supply of healthy mitochondria. In one study, they co-cultured T cells with these donor cells, allowing for a natural transfer. The resulting mito-boosted T cells had enhanced energy production, multiplied more effectively, and showed vastly superior anti-tumor activity. They were simply more resilient and resistant to exhaustion. Another technique, called mitochondrial transplantation, involves isolating healthy mitochondria and directly introducing them into T cells to rejuvenate them. A second major strategy involves nanotechnology. We can use tiny nanocarriers, like microscopic delivery trucks, to transport helpful cargo directly to T cell mitochondria. While delivering whole mitochondria this way is still in early development, a more established approach is to use these nanocarriers to deliver drugs that boost mitochondrial function. For example, nanoparticles can be loaded with compounds that trigger the creation of new mitochondria, or with antioxidants that protect them from damage. By decorating these nanoparticles with molecules that bind specifically to T cells, we can ensure this support package gets delivered right where it's needed. The third and perhaps most powerful strategy is synthetic biology. This allows scientists to rationally re-engineer the internal circuitry of T cells to make them more robust. One method is to genetically engineer T cells to overexpress key metabolic regulators, like a protein called PGC1-alpha. This drives the production of more mitochondria, promoting the formation of long-lasting memory T cells with better anti-tumor activity. Another approach is to create smart T cells with synthetic genetic circuits. Imagine a T cell that can sense the hostile tumor microenvironment and respond automatically. For example, it could be engineered with a synthetic receptor that, upon detecting an immunosuppressive molecule, triggers the T cell to produce its own counteracting agent. These bioengineered T cells would be far better equipped to survive and thrive in the metabolic war zone of a tumor. These strategies aren't just theoretical. They have the potential to significantly enhance our best current cancer treatments. Take CAR T-cell therapy, a game changer for blood cancers where a patient's T cells are engineered to recognize cancer. This therapy has struggled against solid tumors because the carrot cells get exhausted in the harsh tumor microenvironment. By supercharging these CAR T cells with healthy mitochondria before giving them to the patient, we could create a more resilient and persistent fighting force, potentially making this therapy effective against solid tumors for the first time. It could also help with another major treatment, immune checkpoint inhibitors. These drugs work by releasing the brakes on the immune system. However, many patients don't respond, and we now know that mitochondrial dysfunction in their T cells is a key reason why. If the T cells are too metabolically exhausted, releasing the brakes does nothing. By combining checkpoint inhibitors with therapies that boost mitochondrial health, we might be able to turn non-responders into responders and achieve much more durable victories against cancer. Of course, the road ahead has its challenges. Manufacturing these highly specialized cell therapies is complex and expensive. Ensuring the quality, consistency, and safety of the final product is paramount. We need to fully understand the long-term effects of transferring mitochondria and address the ethical considerations that come with manipulating fundamental components of our cells. However, the science is moving at a breathtaking pace. The convergence of immunology, cell biology, and bioengineering has brought us to a new dawn in the fight against cancer. The humble mitochondrion, once just a powerhouse, is now understood to be a master regulator of our immune response. By learning to bioengineer this remarkable organelle, we are not just creating a better drug, we are empowering our own immune system to win the war against cancer from within, bringing us one step closer to a future where cancer is a manageable and ultimately curable disease. Thanks for listening to G Fun Facts Online.